Okay, Rick, I think we're live.
We are indeed.
Okay, so this is both familiar and new to us because I think Rick and I are used to being in front of our exchange audience and doing our welcome, but we're not used to being in our homes. Now you get to see where we live, it's our natural habitat. And we are very grateful that we still have exchange friends. I'm sure you, like we, are living our lives in slightly different ways. If you hear my dogs barking, and I guess you have a dog that could bark,
And a baby.
I don't have that. Although my son has returned home with a horse, no explanation required at this point. But we are very happy that you're still joining us in a virtual way. And we're going to do our usual thing that we always do, which is to kind of start by explaining a little bit about the Science and Entertainment Exchange and about the National Academy of Sciences, our parent institution, because it has never really felt more necessary. An institution like the National Academy of Sciences was kind of built for times like this. I think it was the original vision of Abraham Lincoln, which is how we were created by in his world. He believed that the country should be steered by people who understood science and could chart a better course for the nation by turning to scientists. And I think it's in times like this that we've all thought about ways that we can turn to scientists for good advice. And so when I tell you that we are as busy as we have ever been, and that Rick and I it somehow felt for a few minutes that we might not, God, what will we do? We have been working round the clock because the whole institution is very focused on ways that we can serve the nation. And we have three people here today that are joining us for this event that are very much part of how the institution is responding and how science in general is responding. And so I think that we wanted to be part of that effort, and we wanted to be able to play a role in being able to give something back to our community by doing that. And thank you for giving me a reason for the first time in 3 weeks for putting on mascara, because I haven't really felt the need to do that, but today I did. And Rick and I also figured that with nobody to give us any visual cues as to whether we're actually being interesting, we're going to think that we're amazing today. That'll be the feedback we're going to give each other. And so I'm going to turn it over to Rick to talk a little bit about what the exchange is doing these days, and how busy we are and what we generally do, and to hope that my dogs will remain quiet for a little while longer.
So it's always my job to give the credits before you guys actually get to see the movie. So I just want to say thanks, especially to the AV team who scrambled to make this happen. Amici and David, thank you. And also to the rest of the Office of Communications that basically I was like, what are we going to do? We do events and now we don't have any events. What's going to happen? We're just like, we got this. So Courtney and Jeff and Sachi, thank you. So for those of you who don't know, the Science and Entertainment Exchange is a program of the National Academy of Sciences. We do events that do outreach to Hollywood professionals, writers, producers, studio executives, that kind of person. Now we're online too. But we also have a hotline. There's a real phone number that you can dial that's 844 need sci. That's 844 need sci. If you dial that number, we are still open. It's a phone. We can physically distance and still help you out with a project if you have a feature film, TV show, or video game. We've done over 3,000 consults at this point, and a few of those titles include Avengers: Infinity War, Man of Steel, Black Panther, Captain Marvel. Last but not least, before we get to the meat of this whole thing, I'd be remiss if I didn't thank our sponsors: Alfred P. Sloan Foundation, Howard Hughes Medical Institute, Walt Disney Company, Film Nation, Google, Lyda Hill Foundation, Corteva, and all the individual donors, especially the ones who gave right now for this event to happen. You guys are amazing. We didn't actually even, we weren't sure. So thank you for tuning in and for giving and for everything. So one of the things that is a little bit different about this meeting or this event from other events that you may have experienced around the Science and Entertainment Exchange, you can ask a question at any time, somewhere down here, there's a taskbar. It says Q&A on it. And when you, at any point during the event, not right this second, I'm not responding right this second, but I will in about 5 seconds as soon as I stop talking. I will be responding to folks, and I will also be grabbing your questions for our field experts tonight who are, who really know this stuff. And when we come back on for the Q&A, we'll have a curated list of your questions, of some of the best. And we're all looking at this thing, and we're going to pick some of the top questions and ask to this group. I think we have time for 6 or 7 questions at the end. So without further ado, I'm going to hand it back to Anne so that she can introduce.
And as you know, if you have come to exchange events before, which we presume you have, we don't read CVs. We assume that that part of our job is to bring really smart people to talk to you in events that we produce, and we've done no different in this particular instance. But we did, want to frame this a little bit by saying that we know that there's so much news, so much going on, and sometimes it's hard to filter through all of that. And we were really trying to think about this first event and how we might frame the conversation. And there's been a lot of conversation around vaccines and what that process looks like, and how we might be able to think about that in realistic terms. And there's a whole lot of science that goes into, and there's a lot of science behind that process for vaccines. So we really wanted to start with a conversation that in some ways felt a little bit hopeful, especially at a time that doesn't always feel that way. So that's partly how we thought about this event. So thank you very much for being here. And I'm going to turn it over. And Maria Elena is our first speaker. We put her in the difficult position because it's as I say, it's hard when you don't have an actual audience in front of you when you're doing this. But we're going to bring Maria Elena in as our first speaker, and she's going to get 10 minutes. And she's coming to us from Baylor College in Houston, Texas. And we're very glad that she's here. We're glad that all of our speakers are here. So thank you very much. And I'm going to set your timer and let you go from here.
Excellent, thank you. It's going to be really great to speak to the screen and see actually my face and maybe Rick's face too. But hello, everybody. I'm really, really excited to be here with you all. I actually see there's 485 participants, so that's really excellent. This, I think, as you heard, is the first attempt to launch this virtual reality experiment and start to walk you through what goes on, an inventor's mind during this indeed long and complex process of what it means to develop a vaccine. It is really a real honor, and you will hear from the couple of panelists after me. They're, believe it or not, they're my greatest heroes, Dr. Jerome Kim and Dr. Jeffrey Kahn, because they really will describe to you, after what I'm going to try to set a framework, is really how vaccine development does follow actually a pretty systematic approach. But this approach actually has to be embedded into a very complicated societal ecosystem. And I think at least it has to take into consideration 7 types of relationships, right? So it's not only the scientific framework of how you develop a vaccine or the biological basis. You have to consider a lot of clinical implications, behavioral, societal, operational, environmental, economical, certainly ethical, so it's a very complex ecosystem. But what it's very important to keep in mind is that it requires to be done in community, or what we call partnerships, and is no longer anymore a single group or a single organization. And Dr. Kim most likely will elaborate this, because his vaccine center, as well as ours, operate under this concept of product development partnerships. And second, it certainly has to ensure that by developing vaccines, we do not cause unintended consequences, leading to either wasted time, effort, resources, and specifically Dr. Khan will elaborate on this. For example, we certainly do not want to develop a vaccine that has no scientific feasibility, or that there's going to be no demand for it, or that it actually can cause harm, or that can even be prohibitive with regards to the cost, and that the population are not going to really even be able to access it. So I want to lay the playing field before I go into how exactly gets done. So the reality is that we can divvy up the vaccine in three different types of business cases, and three different types of value propositions of these vaccines. So vaccines can be intended primarily for what we call dual markets, for high income markets, low middle income markets, most likely these are what the vaccines that are licensed look like: hepatitis B, human papillomavirus. There's also vaccines, and that's usually what I work very heavily on, is vaccines that are intended for only low middle income countries. And these are really hard to develop because, as you know, they already come with this preconception that there is no market. But the reality, in fact, that there's a huge potential public health market. And that's like malaria or these, parasitic worms and other kind of interesting neglected diseases that we call. And then today, I think we want to also focus your attention on how do you develop a vaccine against an outbreak, an Ebola, or now a COVID-19. These are also quite hard because you're developing against a pathogen that you probably don't even know what it is. And even if you have something that you think that could resemble, but if you're really going to prepare a vaccine anticipating a outbreak or a pandemic, you have to really be quite clever. And I think that's exactly what this title of this event is, right? How can you promise something that can lead to peril because you maybe can never develop it, but at the same time you have to make decisions so fast that you may not be making the right decisions in the step. So with that brief, setting up the playing field, I do want to focus on the components of developing a vaccine that really are the early scientific steps. What happens from the laboratory bench, from that, innovator, scientist head, discovery, creation, I guess that the actual product has to be designed, and how that eventually will lead into the bedside. And this is actually a step that everybody calls transitioning through the first valley of death. I actually don't think that sounds quite optimistic. It's quite dire, right? So I really always try to use optimistic words. So I'm going to tell you that we are, going to talk about transitioning through the valley of hope. So we scientists are really artistic like you are. We create things. So I'm going to use my example. I love cooking, or at least I used to much more before. I'm Italian. I'm also Honduran, so my roots clearly have some sort of artistic ability of cooking. So what I'm going to do is I'm going to give you my recipe of how do I make a small lot of a vaccine. It will serve around 100 to 500 people, and I'll probably take me around 18 to 36 months to, accomplish this recipe. Here's the ingredients. You need people, right? The appropriate people with the right capacity, usually with a lot of different scientific disciplines working together, and certainly you need also a second ingredient, an infrastructure, whether it's an academic-based institution where all these innovative ideas arise. But most importantly, you have to have a robust pipeline of candidates. This is one thing that we're very clear. Vaccines tend to have a very high rate of failure. That's therefore why the valley of death. And so we need to make sure that we start with a lot of things. We need models so that we can test our vaccines, whether they're in vivo or in vitro. And of course, we need money for us to be able to do this. Just to give you some numbers, usually from the preclinical to the clinical stage, there's around 40 to 50% success rate. Only 10 to 13% actually go even beyond there. You probably need around $6 million to $28 million just to get to that. Now, the steps. I have 3 minutes left. So you select a disease or a pathogen of your choice, and you pretty much do like every business. You do a SWOT analysis. You look, is it feasible? How much is it going to cost? Are there competitors? Does it make sense scientifically, technically? You then have to design what we call the product development strategy. That's where it gets really tricky. So select a pathogen. How do you decide what antigen to develop your vaccine? You have to really look for something that you can disrupt as an essential function within the pathogen. For example, blocking a virus to get into the cell, blocking the feeding pathway of one of these intestinal worms. We look at therefore then their genetic code and find this candidate so that we can actually design it. And you can use all these omics, genomics, proteomics, transcriptomics. We use that DNA, and then we have much more complicated of, are we going to do a vaccine using the DNA, the RNA, the protein? Are we going to package it as a protein, as a live virus, as, you know, as a particle, as a viral-like particle? So there's so many things that we need to kind of design. But more importantly is the functionality, right? So again, here is when you deliver it, are you inducing a response that is going to protect the individual, that is not going to exacerbate that response in such a way that causes adverse events? Does this, whether it's an antibody or a cell, is this going to really protect the individual when you get hit with the actual pathogen? Do you want 100% protection? Do you want partial protection? How much is it going to cost? Is it going to be very expensive to use this platform? Sometimes we use yeast to make the vaccines, bacteria to make the vaccines. We've been hearing a lot about RNA-based vaccines. Can everybody make it? Can you scale it? Can you make them easily reproducible, that everybody can make it? Can the world have access to them? So again, I leave you with that, with this valley of hope, which is really where the inventor minds in mostly academic institutions, the creativity starts. Unfortunately, can be quite daunting because we scientists are sometimes not very focused. And so we want to try to do absolutely everything to learn about the pathogens, learn about, how we can tweak and tinker the vaccine. But if we're really serious, then it would be handed over to someone like, for instance, Dr. Jerome Kim, who now in the next 20 seconds will be able to describe to you the next step. Thank you for your attention.
So that was I don't even have to do a job. You've done my job. Thank you very much, Maria Elena. That was wonderful. And I should point out that our next speaker, Jerome Kim, gets the special Valiants Award because he is in South Korea. And you can do the math on the time zones here to know that this is very, very early in the morning for him. So he is not wearing his pajamas like probably some of those who are tuning into this, but he would have every excuse to do that. So I hand it over to him with great thanks for getting up so early in the morning, before his 5 a.m. run, in fact, to be here with us today. So thank you very much, Jerome.
It's my pleasure. Good day to everybody. And I have the really interesting topic of talking to you about the vaccine industry and how they do things. So I hope it's not right after lunch for most of you, but vaccine development, actually, we wouldn't have the vaccines we have today if it weren't for the guys in the gray suits who translate the what Maria Elena and the scientists do in the laboratory into products that we can put into people and get countries to take up. And the decade of vaccines maybe saved 2.5 million children a year from implementing the vaccines that we have. So those people who manufacture the vaccines are really doing a tremendous amount of good around the world. Maria Elena actually had talked about this a bit, but under normal circumstances, the vaccine industry tells us it takes 5 to 10 years and about $1 billion to develop each vaccine. And that's from the time it's in the laboratory until the time it's injected into your child in the doctor's office. And as she pointed out, only 1 in 10, actually 93% of products fail to go all the way from the laboratory into the doctor's office. And so that's kind of the background against which a lot of the vaccine manufacturing decisions are going to be made. Now, I think everyone recognizes that COVID-19 does not fit by any measure under normal circumstances. So I'll also go through briefly the accelerated process that is going to be needed in order to try to accelerate the development of a COVID-19 vaccine. So Maria Elena actually went through the mad scientist moment, which is the one that we see in the films that you know, where there are people with test tubes and you know, rats and lab animals and you know, fancy structures that rotate on a screen. And I can tell you, Dick Cheney really loved that when he went to visit the Vaccine Research Center at the NIH, because you can look at the outer surface coat of a protein, rotate it, turn it upside down, backwards, and then talk about how the vaccine will develop these antibodies. It will bind to and neutralize the proteins that are shown on the screen. So, you know, all of that is great. But that happens at the front end. And what I'm going to do is really talk to you about what happens in the middle. So once you have the vaccine and it's coming out of the laboratory, it has to make it over the first gap. Maria Elena called it the Valley of Death. You can call it the translational gap. That is translating it from the university lab or the lab at a big company or a biotech into formal testing in humans. So, you know, the laboratory itself has probably tested the vaccine to see, you know, injected it into a mouse and saw that the vaccine makes the right protective or immune responses. You can actually then take it into larger animals. You know, sometimes, for instance, influenza vaccines are tested in ferrets. Often the vaccines are tested in monkeys because, you know, monkeys are the closest to humans, and we really want to know that if we inject this vaccine into a human being, that we're going to get a response that is something we expect and something that protects. And so often this series of animal, you know, larger and larger animal studies goes on, and it takes months, sometimes years, in order to get through this process, because at the same time, the company is tweaking the design of the vaccine, going back and forth to say, well, if we add this or take out this, what will we get that's better? And it's a process, I think, as it was described previously. Once you have the vaccine that you think you want to take into humans, you have to actually make it in a particular way, because that particular vaccine that goes into humans now has to pass through a series of regulatory hurdles, which are established in order to guarantee that when it's injected into humans, it's safe. And that's you know, we call them toxicology studies. They're often injected into at least 2 different animal species, and we look to see if the vaccine actually causes any harm by itself in those animal species. So that's another 6 months. And then it goes into humans in what's called a phase 1, or sometimes a first in human study. And the real purpose of that is to make sure that it doesn't harm people. And you get some indication that it's making the right protective responses. So again, vaccines make protective responses, which we call immune responses. And the phase 1 is really the best way to look at that. Now, if the phase 1 looks good, then the company says, oh, okay, let's move this on to the next stage, which is phase 2. It's very logical. And phase 2 is actually a much larger study. So phase 1 may be 50 people. Phase 2 could be 200 people. And it really begins to explore things like, what's the right dose? Or what's the right dose in a pediatric population? I mean, these are the things that the companies have to do. And often phase 1 and phase 2 are sequential. So you do one, you analyze the data, you move on to the next, you analyze the data. And if phase 2 works and you have the right immune responses, then you move to phase 3. And phase 3 is the big one. That's 1,000 people. The phase 3 trial we did in Thailand was 16,000 people. The Army did another phase 3 trial in Thailand to look for hepatitis A vaccine. That was 43,000 children. So these are huge studies, but they really are intended to prove beyond the shadow of a doubt that a vaccine is safe and effective and prevents the disease or infection. But once you have phase 3, so say the vaccine now works, it's not like you can just start selling it. Now it has to go in the US to the Food and Drug Administration or the EMA in the European Union, and they take a long time to review hundreds of thousands of pages of documents that go through how the trials were done, what the data are. There are often questions that they have, and the manufacturers have to answer all of these. And this process takes months to a year sometimes in order to go through. And you want the FDA to make sure that things are safe and efficacious. Once the FDA approval is given, you can actually start selling your vaccine, but who's going to use it? So usually there's another committee. In the US, it's called the Advisory Committee on Immunization Practice, ACIP, and they make a recommendation to use the vaccine in children of a certain age, for instance. And then the companies can stop marketing it, assuming that they have manufacturing. So manufacturing is the next step. And probably if you've never visited a vaccine manufacturing plant, you should. Probably the closest thing in my mind is if you go to one of these brewery bars and you see these giant tanks. It's a lot of stainless steel, a lot of pipes, and vaccine is being moved under sterile conditions from one room to another. And it's all done under this very rigorous international code of practice called GMP. And that ensures that the vaccines that go into people are going to be done at a high quality so that the vaccines give you reproducible protection in human populations that they're tested in. And often it takes over a year to make this manufacturing plant. So when Sanofi was rolling out its dengue vaccine, it had already paid for a large manufacturing plant just to take care of that particular dengue vaccine. So this is a very costly enterprise, and the companies don't commit to them unless they're absolutely sure this vaccine is going to go forward. They want it to be ready. They want production to be ready for approval, so that as soon as it's approved and gets a recommendation, it can be sold in large quantity. So on the world stage, the FDA is important, but the thing that helps a developing country like Ghana or Tanzania make a decision to use a vaccine is actually a different group called the WHO Prequalification Group, which does the same thing as FDA, and a group called SAGE, which, like it sounds, is a bunch of experts. They look at the data and say, you know what, we should use this vaccine in global health. And that recommendation is critical because many of the smaller countries don't have the capacity to review the documents and make recommendations. So they depend on the World Health Organization for information. And then there's a separate organization called Gavi, because many of the countries can't pay, as we do in the United States, $200 for pneumococcal conjugate vaccine per dose. The Gavi price is $3.10. So the countries and organizations that donate to Gavi subsidize these vaccines for global health. And so you can imagine a COVID vaccine, we need at least 7 billion doses. And countries around the world, in the poorer parts of the world, aren't going to be able to pay the full cost for the vaccine. But we know, for instance, for COVID, that we have to do it. So how does COVID do it? It's a truncated, accelerated process. It was announced initially in January 2020, and by March 17th, we had a vaccine tested in humans. And this was made possible, this acceleration of the phase was made possible by several things. There's an organization called CEPI, the Coalition for Epidemic Preparedness Innovations, that has a $1 billion fund to make vaccines for outbreak diseases, and they were ready for this. So they made an announcement, picked 4 companies right away. They're now 8. And the first of those companies, Moderna, which is a US company, injected the vaccine with the US NIH into volunteers roughly 8 weeks later. Really amazing. The other vaccines trials should start in April. Again, very truncated schedule. And so they'll move through the process, phase 1, phase 2, phase 3, at maximum speed, which may mean that by the late winter, they may have a vaccine proven, and that's Dr. Fauci's estimate, 12 to 18 months. So those are the usual and truncated forms of vaccine development. Thanks.
Thank you very much, Jerome. You can see that we are putting together a pretty complete package here, but some of you, if you have been to exchange events before, may recognize both the name and the face. Oh, and there go my dogs. Thank you very much, right on cue. Jeff Kahn has done events with us before. And so from our point of view, it really wasn't going to be a complete conversation without thinking about some of the ethical issues associated with this. I think that we got a really good sense from our previous 2 speakers of sort of the peril and the promise, but there's also the people involved in this and how does this impact individuals. And I think that when we talked to Jeff about this, it became very clear that we needed to have his perspective on this, because I think that we have to think about this in very broad terms. So we were very glad that Jeff could also join us. He didn't have to come from as far as South Korea. He's in Baltimore from Johns Hopkins. And I know a lot of people are probably looking at that Johns Hopkins model that so many of us are clicking and refreshing every day to see the numbers. So thank you, Jeff.
Thank you, Anne, and thank you, Rick and Jerome and Maria Elena. It's been a great conversation so far. Thank you for having me. And welcome everybody from here in beautiful Baltimore. So I'm going to talk about the ethics of human participation in vaccine trials. I'm going to focus on 5 different points. The first is when, and you heard Jerome talking about a truncated, expedited process. So there's an important question about the ethics of moving for the first time into humans. And there's usually a process that includes, as you heard Jerome say, multiple stages in which different animal models are used. And that's to both make sure that it's a safe vaccine and it has some effect on creating antibodies so that it will be effective when it gets into humans. In a truncated model, as Jerome mentioned, there's less time and probably in fewer animal models. And so that raises some alarms from an ethics perspective. It's important, and in the COVID outbreak, it's sort of obviously important that we need to go quickly. 18 to 36 months is too long. Even a year seems, and 18 months seems like a really long time in this context. But we don't want to go into humans for the first time until we know that it's safe. So the when question is really important. As Jerome said, there's all sorts of expert panels that are trying to decide how fast is fast enough, but not too fast to create too much risk for the people who would be injected with these experimental first in human or very early phase vaccines. The second of my 5 points is who? Who are the people who would be the volunteers who would participate in these early and later phase trials? And the answer is they're volunteers, and they need to be healthy volunteers. So when you think about clinical trials, sometimes you think about people who are sick and they're participating in a clinical trial for a new way to be treated for whatever it is that is making them ill or the disease that they have. That's not the way it works for vaccines, because you have to have somebody who has not been exposed to the thing you're trying to vaccinate them for. They must be healthy volunteers. So you're asking healthy people to volunteer to undertake risk of some kind of potential harm based on the injection of something that has not been tried in humans or only has been tried in very few humans. So it's about safety for the people who are healthy volunteers who probably at the early stage almost certainly will not receive any benefit from that participation. So as Jerome said, the first phases of trials are about safety. Are they safe to put into people? Nothing about whether they work, whether they have efficacy. It's all about safety. So it isn't about benefit to the people who are volunteers. It's only about safety to them. And we want to make sure it's as safe as we can predict before it goes into humans for the first time. And in these truncated models where the time is sped up, we have to be really clear with people that we only know what we know, and we want to make sure you understand that before you put yourself in harm's way. So that's number 2, who. The third is how. How would this work? So once you inject somebody with a vaccine, and it's the later phase trial, so we're talking about knowing that it's safe enough to move to the next phase, and that usually means that the dose is higher in the later phases of a vaccine trial. You would want to know whether it's efficacious. Does it actually do the thing you're hoping it will, which is protect people from infection? So how would you know that? You would need for them to be first vaccinated or injected with the thing you hope will vaccinate them. And then you need them to be exposed to the infection that you're trying to protect them from getting. So there are 2 ways for that to happen. One is called the challenge study, where you intentionally expose somebody to the thing that you're trying to protect them from. So you would inject them, or in a case of COVID, maybe give them some kind of nasal spray because of the respiratory nature of the infection. An intentional exposure experiment is sort of a very big red flag, again, because you're giving somebody an infection intentionally. You hope they'll be protected, but you don't know for sure. So the other way that it happens is you let people go about their business and they get exposed through the course of their activities. That's probably how it would work in the case of a COVID vaccine, I'm thinking. And I think as the planning has progressed, that's what the process will be. The hope is that the vaccine will provide either some or full protection, and in the exposure, the person won't get sick. But of course, we don't know that. And so in the process of recruiting people, we need to tell them that you're going to be injected with something that's experimental. We may not know whether it's even safe yet, but maybe we know it's safe, and we don't know whether it works. And when you go out and potentially get exposed, you may actually be protected, but you might not. We don't know which of those things will happen. We don't want you to bank on the fact that you're getting protection when we don't know whether that's true. So that was 3. Number 4 is why. Why would people participate in research of the sort we're describing here. So what's their motivation? And how would we make sure they understand what they're getting into in ways that make it acceptable for them to participate? That's called the informed consent process. So the right answer from an ethics perspective is, I want to do this to make sure that this is safe for other people who will come after me. I realize it may not be beneficial to me. If it is, that's a kind of good and lucky outcome and consequence, but that's not the motivation for my participation. Certainly in an early phase trial, that can't be the right motivation. And I hope it will benefit the people who will come after me, society at large, the future. That's an altruistic motivation for something that doesn't benefit me at all as an individual. But people may also think, well, maybe I'll get some protection from this infection if I participate in this trial. That may be true, but it may not be true. So we don't want them to be making their judgment about participation solely on the hope that they will receive some benefit. And then the third thing is, in trials that involve healthy subjects, we often pay them. We give them money. And we want to make sure that that compensation isn't so much that they're doing it for the money and not for the good motivation, the right motivation. So we want to give them money to compensate them for the time that they take, or the having to give time from work or travel to and from the place where they will participate, but not as a way of sort of paying them to accept risk. So the current trial that's now just started, the Moderna trial that Jerome mentioned, has just started to enroll subjects. They're being paid $1,000 over a year-long participation, with $100 per visit being the total amount of payment. So that doesn't seem like a lot. It seems like an appropriate amount to compensate people for the time that they'll be spending in trying to help society beat this disease. Number 5 is another how. And let's assume that we get to a point where we make it through all these phases of a trial, and there's an effective vaccine that we maybe actually can afford, that isn't so much that it's unaffordable, and we figure out how to pay for it. Then there's an important ethics question about how we allocate that, especially in the early stages of the production of a vaccine, when we can't make enough for everybody who would benefit from it. And we have to decide who should get it first. And the answers tend to focus on those who are at greatest risk from the infection and those who occupy critical roles in society. So first responders need to be protected. Those are the people who are responding when you call 911, ambulance, EMT, fire, police. And then the frontline healthcare workers who are, of course, caring for people who would be infected. So those first responders generally get to the front of the line when we talk about allocating something that we don't have enough of in the context of a new vaccine. Same arguments actually happening in the context of some of the shortages showing up in the hospitals where we're treating patients with COVID. The second category would be people at highest risk within the population. So it depends on the particular infection. So as we've heard, the older population seems to be at higher risk of very serious consequences of COVID infection in this particular outbreak. Younger children less so. So we would imagine that you would vaccinate older people first and younger people later. So first responders first, those at highest risk from the infection second, and then maybe third, think about the people who occupy critical roles to keep the infrastructure working in our cities and towns and states and federal government. So they're not first responders, but they're the people who are making sure that there are groceries in the grocery store. They're critical infrastructure, critical and essential workers, but they're not maybe at immediate risk of the bad consequences of the infection. So in the order of highest priority, the people who are responding to the emergency nature of the outbreak. Second, those who are at highest risk from the infection. Third, the critical and essential workers, and then comes everybody else. And we hope we can get to the everybody else as quickly as possible, ramping up production to make that possible. So with that, I will stop, and I think it's time for question and answer. Thanks for the attention. It's been a really interesting conversation so far. I look forward to more.
Well, Rick and I want to thank all of our speakers. Really, you all have been wonderful, and especially wonderful because you did this as our very first virtual event. And we tried to set the bar, the technical bar, fairly low, because Rick and I did not consider ourselves to be high jumpers in this situation. But you all came through with shining colors. So thank you very much. And it was a really interesting and important conversation. But our audience has also been terrific. We have been watching the questions come in, and there are a lot of them. We will not get to all of them by the look of them, because you guys have been really active, and we genuinely appreciate that. So Rick, with his discerning eye, has been very carefully monitoring all those questions, and he's going to be a little bit, I guess you're going to be the man behind the curtain now, and you're going to throw them out there to our speakers, because you've been keeping track. I know you've been keeping track.
So I think the first question, which is a bit of an amalgam. Oh, and we need to have the speakers unmuted.
Yeah, we want our speakers back, because we can't answer any of these questions.
Yeah, I don't know the answer.
We can't answer any of these questions.
This question is an amalgam of a few questions, but I think I'm hitting a couple of people, what a couple of people are asking. Is COVID-19 mutating? And if so, how does that affect the approach with a vaccine?
Should I take the first stab, at least from the laboratory perspective? So again, you're right, and and if the discovery really comes from looking at how the genetic coding is changing currently in this virus as an example, what we do is again use this combination of disciplines, whether it's structural biology or genomics, and really try to see if we can design different potential, again, candidates. In this case, we know that we've been focusing on that spike protein that the virus uses, and us specifically even focus even more in detail the domain that spike protein uses to bind to the receptor in the humans. How much is that being mutated? Is it conserved? Is it changing the structure of the virus, making it more affinity to the receptor or less? And then ideally, you're right, the hope is that these types of outbreak type of vaccines are going to be rapidly switched or even made as universal, where maybe even the SARS vaccine could confer some protection against the SARS-2, maybe even the SARS-2 or COVID-19 vaccine can confer protection towards a potentially upcoming new coronavirus that's going to show up. We are trying to do that also not only with COVID-19. As you know, there's also big efforts with regards to influenza that everybody's trying to look to see how we can improve and not have to every time start from scratch. So it's a lot of scientific information in the lab. And I'll throw it over to Jerome and Dr. Khan.
So I can do the next part, I think. So my lab used to do sequencing before I went to South Korea. So we've been looking at some of the information that's available around the world. There have been about 1,000 now, probably a little more than 1,000 full-length genomes of the virus sequenced. So 30,000 base pairs, so those little RNA nucleic acids that are strung together to make the viral, what makes the virus the virus. There have been only 11 point mutations in that time. In the 1,000, so remember, we've only got 1,000, and there are over 300,000 infections known in the world. So we don't have a big picture yet. But in the picture that's appeared, the virus doesn't appear to be mutating that quickly. In fact, probably 8 to 10-fold less mutations per year than you would see with influenza virus. So that is somewhat hopeful for vaccine development. It may mean that the current crop of vaccines, which are based on the original viral sequences, hopefully will continue to protect.
So let me ask a question to both of you, because I'm not a scientist. I studied microbiology in college, but that was a long time ago, 1980 something. I won't tell you the year. As viruses mutate, do they evolve to become more virulent or less? I mean, this is an interesting question for the entertainment folks in the audience, because in movies you often see, Contagion is an example where the virus, as it mutated, got more virulent and caused people to die in ways that the early strains did not. But that isn't really how virus biology tends to work, right? As viruses evolve, they don't become more virulent as a matter of course. Maybe you can speak to that.
Want to start, Jerome?
Okay. So I think actually Maria Elena made a great example. The original SARS virus, SARS-1, back in the early 2000s, is about 80% similar to the current virus. And those changes, those 20% of the genome which has changed, actually has conferred a very different disease. So in SARS-1, remember that the people who were transmitting were very sick. So it was actually kind of easy to put them in, isolate them because we knew who they were. With SARS-2, now just 20% difference in the sequences, you're getting a disease where 80% of people are asymptomatic, but can spread. And so that change in the biology conferred by 20% change in the structure. I mean, the viruses look very similar. I mean, in fact, we're working with Dr. Bottazzi on a vaccine together, and the areas of SARS-1 and SARS-2, even though the sequences are different, the structures look the same. And so, the different monoclonal antibodies, and again, this gets to the structural biology thing, look like they would bind in exactly the same place. So there might be cross-neutralization and things. So it's really fascinating. And the same would be true, for instance, of Zika and dengue. Same distance, very different diseases. One causes these birth defects. The other causes, primarily hemorrhagic fever. So again, 20% difference in the virus sequence. So I think that gets to part of the question, and also the question around will a vaccine for one protect against the other? It's very interesting. Sorry, Maria Elena.
I agree. And I think indeed you have to also look where these differences are occurring, and even and are you designing therefore a vaccine that interferes something that clearly is being protected by the virus? Because again, the virus needs to make sure it survives. So that's where you want to hit them hard, right? And they may be changing all these other things because it gives them advantage, but you really want to block something that it is quite essential. And we know that it is essential to cause disease to introduce themselves into the cells, and that's, in this case, that's the approach that everybody's trying to block.
Yeah. So viruses want to infect and allow themselves to be spread to others, right? So killing the host quickly is a bad idea from a virus's perspective.
Correct.
Which is an interesting way to think about sort of virus biology. Anyway, Rick, we probably should go to the next.
So do you think it's possible that, which scenario do you think is more possible or more likely, that the vaccine will not be owned by a specific country or corporation and will be available widely to anyone who needs it? Or do you think the more likely scenario is that the world's poor will not be getting the vaccine?
Jerome, I see you're going to start.
So I think, again, it depends on who's funding the vaccine, right? I think if the US government is funding the vaccine, then there will be a focus on protecting US populations. That's the remit of US government agencies. For those countries that have committed to CEPI, CEPI has a series of access agreements in place that are supposed to ensure that a vaccine, when it's available, can be widely distributed throughout the world. And so I'm not party to any of the CEPI agreements, but that's the way it worked in theory, because the groups, the Gates Foundation, the Wellcome Trust, Norway, Sweden, Germany, Japan, India, they all have an interest in making sure that vaccines that are developed through the CEPI mechanism really are provided to people around the world. So I think, let's hope that this mechanism proves to be the kind of thing that we need in order to ensure that a global prioritization can occur, as Jeff described, because that's going to be really important. Access to, a vaccine that isn't used is useless, no matter how effective it is. And the other thing is that COVID, if we let COVID rage unchecked in Africa or in South Asia, we'll never be free of it, because it can always come back and cause a problem elsewhere.
I would just say to your point, Jerome, this is a test of the system that CEPI is meant to represent, right? This is the first time we've seen this kind of situation, and it's exactly the reason CEPI was set up. So, this will be an opportunity to see whether the way it's been organized and funded will work the way it's supposed to. And if it doesn't, there'll be, I'm sure, a lot of conversation and hand-wringing about what to do.
I mean, if I may also add a little bit, and I mean, it clearly it's not been the first time, right? That, we, those of us who are in the field of developing vaccines, whether they are for, again, diseases for high income combination, again, like I mentioned, I also work on diseases that are very specific for the low middle income countries. I think there is not one cookie cutter made model, but the idea is also that, whatever technology is being used, whether it's being developed by a small biotech that eventually is going to be purchased by a government, or even sold directly by the companies, there has to be a framework where, again, they can, one, be easily scaled up such that, there's not going to be ever a single producer for all the vaccines that we need. So that's why there's networks of vaccine manufacturers, including the developing country vaccine manufacturing networks. Then there's definitely, like Jerome mentions, there's Gavi that clearly focuses on access to governments to be able to can afford them, but then be self-reliant and sustain that process. You have certainly the PAHO, Pan American Health Organization revolving funds, UNICEF. So there are many mechanisms. The only concern, of course, is how quickly you can achieve those equitable, sustainable, highly, reproducible, scalable, processes. But I mean, I think it can be done. We'll see what happens. I agree with the CEPI framework.
And I think that the flip side of this is, if you look at a vaccine used in the United States, like all of your children have gotten rotavirus vaccine, it's 80 to 90% effective. Perfect. It was approved in the US in 2007, recommended by WHO in 2009, and now in 2020, still only 20 to 30% of the children in the world have received all of the doses that they need, even though it's been officially approved in over 80 countries. So there are inequities in access that occur in the normal setting that I think, for an outbreak disease like COVID that has dramatic implications for global health and populations, we're really going to have a better mechanism than the one that exists now. And we hope CEPI will do that.
Well, and the interesting thing, the last thing to say about this, there's huge private funding that is stepping up to help here, which is, again, in a way, a kind of warning, right? Why is it the Gates Foundation and Wellcome Trust are the people who are stepping up? It's great that they are, but this is a governmental role in large part. And you mentioned Norway, which I think is in some sense the outlier rather than the most common example. And so how are we going as a world collective to confront these kinds of challenges if the countries themselves don't step up?
So we have a few questions around what we currently have on the table to treat COVID. People are asking about some malaria medications and also whether or not there's some degree of immunity from other vaccines. Does anyone like to weigh in on that?
Well, maybe I can just comment. I mean, of course, vaccines, I think, as you have already understood, is to prevent actually to even have infection. So, arguably given to healthy populations. Indeed, it is going to be a long-term solution for not only this, but eventually future. And that's why for vaccines, you definitely, have to have these programs quite sustainable, because you're eventually going to want to use them in healthy populations. Definitely the urgency right now is also to make sure you treat those who unfortunately are symptomatic, and clearly even those that are severely with severe symptoms. So I appreciate that everybody's trying to repurpose and say, well, maybe this will work. I used it for this thing. It's like me telling my dad, my friend told me I should take this medicine because it worked for him for this. And I'm like, Dad, I mean, you can't just automedicate yourself. So we have to also be very cautious that they have to also go through very rigorous evaluations. I know there's, various that are being currently evaluated, including the one you mentioned, the chloroquine with the azithromycin, even transferring convalescent plasma from one person to another. So clearly, of course, we have to look at all these strategies, but they have to be in the context of being under good ethics, under good regulatory, and certainly with very specific checks and balances, whether you're doing it as a last resource because you're doing it as a compassionate use, or because you're actually physically really doing a statistically robust evaluation, that then you can share the data and use it for more wide range of populations. It's also quite a complex a complex ecosystem in that, and we can also go into diagnostics because I know Dr. Kim would probably also talk about what's going on in the diagnostic arena. But yes, usually you have those three hand in hand, right? You have to diagnose, ideally you want to prevent, but you also have to have tools to treat. And they should all kind of be aligned at some level.
And I think the same process applies. I think that you started to make this point. The same process that we do for vaccines, phase 1, phase 2, and phase 3, actually should occur for drugs as well. And I think there you can go into the ethics of it, Jeff, but I mean, if we say that hydroxychloroquine should be used, then usually the FDA requires that both safety and efficacy be proven. And as Dr. Fauci, I think, has said many times on TV, really we need to study this in a trial where we compare this to nothing. Because if you took 100 people with COVID-19 disease, 98% would get better with just supportive care. So really you want to answer the key questions. Does it decrease the number of people who need ventilators? Does it decrease the number of people who have to be hospitalized? Does it clear the virus from secretions faster, which means more people get out of the hospital faster, which would be great? And then importantly, can you give it to a healthcare provider and prevent them from getting COVID-19 infection? So again, can it be used prophylactically? But these are questions that should be studied, because if a doctor just started taking hydroxychloroquine and azithromycin and it had an impact on their heart, and the two could cause sudden death. And they've seen this now already in several countries. The other thing is about other vaccines. So there's some suggestion that BCG vaccination may somehow protect. Again, this is something that repurposing an old vaccine is like repurposing an old medicine. We really have to be able to show that it has a benefit, that it's not hurting people. And that's something that we should do in studies, not empirically because we think it works, because all of these things can have consequences that are unanticipated and bad.
So we're just about out of time right now. I have one quick question for each of the three of you that I think, again, brings a bunch of people's questions together. For each of you individually, what's a benchmark that will make you feel comfortable being with a large group of people in a contained environment in the future?
I want to be last. I was first, now I'll be last.
Okay, I can do that. So I would say either what we call long-acting prophylaxis, which we're developing now for HIV, where you can get injected, once a month. A gamma globulin-like thing. I think many of you who are older, I guess like me, remember being injected with hepatitis A, prevent hepatitis A if we're going on a trip somewhere where hepatitis A was a problem before there was a vaccine, or a vaccine. Having a vaccine would provide a level of herd protection, would protect you, but also a level of herd protection in the community, hopefully, that will protect other people as well who potentially aren't vaccinated. So that's my quick answer.
And I would say I'd love to know that I had immunity, which would mean I was exposed and had a symptom-free recovery. So that would be, for me, be the best answer. I don't know how we get there. So, I think, Rick, you're really asking, how long do we need to know that there have been no new infections in the community before we feel safe going out? and so it's got to be weeks, if not months, I think, and that's a long time for doing what we're doing now. One last thing I want to say about the antimalarials: there's been a run on effectively hoarding of antimalarials, sometimes written prescriptions by physicians for themselves and their families, which has really caused a lot of problem for the people who need those drugs as a matter of their routine care. People who have lupus and other autoimmune illness. So we at Hopkins and I know lots of other places are restricting dispensing those drugs, believe it or not, because of the concerns about hoarding and inability to have enough supply for people who really need them.
And I think from my side is, certainly seeing this what they're calling, like this reduction of the curves, with primarily seeing that we're getting certainly less and less, severe cases that have to go hospitalized, and of course, serious enough that even we stop seeing deaths. The likelihood is, again, it goes back to Dr. Khan's, that eventually, most likely, there's going to be enough population that are at some level immune. So then we would all feel that there's not going to be that heavy transmission anymore when we kind of like get ourselves again congregated with our family members or or our friends. Optimism, though, there's definitely optimism, and it will happen, whether it's a technology or a successful physical distancing. It will happen.
Well, that's a good way to end on an optimistic note. And so we appreciate that. And we appreciate all of you. And I know that if there was a way to give, virtual applause, you would all be hearing that applause right now, because we know that this was a really great event and really helpful and really interesting. And we apologize again that we couldn't get to all the questions. There were just so many of them. And Rick, you did a spectacular job of selecting from among the many, because there were just a lot. We do want to thank our colleagues at the Health and Medicine Division at the National Academies and at the National Academy of Medicine, because it's all part of the big infrastructure at the academies, and they were super helpful in introducing us to Maria Elena and to Jerome, because we wouldn't have been able to have them here without them. So we want to make sure that we thank our colleagues there. This was a joint effort. And for anybody who is interested, at the National Academy's website, we do have a standing committee that is responding very quickly to a variety of inquiries, and you can find that on the main website at the academies. It's very big and very obvious. You can see that there. But you'll hear more from us in the future. We're going to continue to do events, some of which will be COVID related, but we also want to give you kind of a break from, it wouldn't be all COVID all the time, that there's still a lot of other science in the world. And so Rick and I have been talking a lot about the kinds of things that we can do that are just ways to think about science more broadly.
Yeah, and we're gonna ask you to weigh in on that. So we're gonna send you a poll, and we really, really, really are gonna take this poll very seriously about what we do in the future and how we could do this event better. So if you will fill out that poll, we'd be hugely appreciative. And thank you for tuning in today. We appreciate it.
Yes, thank you. Bye.